We're now moving into the fourth part of our Enterobacteriaceae lecture. And in this lecture, we're going to focus on the organism Shigella. There are 4 species of Shigella that are medically relevant, and these are Flexneri, Sonii, Boydii, and Dysenteriae. The Shigella are very closely related to Escherichia organisms. However, E. coli is part of the normal gastrointestinal flora. Shigella is not. Shigella causes the disease bacillary dysentery. Shigella is also non-modal. That's very important. Many of our Enterobacteriaceae members are modal, such as E. coli and Salmonella, but Shigella is non-modal. Except for some strains of Shigella flexneri, they generally do not produce gas from glucose. They are urea. They don't hydrolyze urea, so they are urease negative. They do not produce hydrogen sulfide, so Salmonella produces hydrogen sulfide, Shigella does not, and they do not decarboxylate lysine. Now, the organism Shigella sonii is a very slow fermenter of lactose. So it will turn bright pink on McConkey agar, which is indicative of a lactose fermenter, after 2 days of incubation. We normally will look at stool cultures after an overnight or 24-hour incubation. They might be put back into the incubator and then looked at again in 48 hours, depending on the lab you're in and what you're looking for. Generally, stool samples aren't kept that much longer because most of the pathogens will grow within 48 hours, unless you're looking at a slower growing organism, and usually those are incubated in special conditions. Shigella does have antigenic structures that are helpful to differentiate these species. So the species are differentiated into 4 major O antigen groups. So Shigella dysenteriae is group A, Flexneri is group B, Boydii is C, and Sonii is D. Several Shigella serotypes exist within each species, except for Shigella sonii, which has only 1. All Shigella possess O antigens. Some strains might produce K antigens or capsules, but in general, they're not considered capsule producers. The K antigen is heat labile and can be removed by boiling the organism. So sometimes you need to boil organisms to detect the O antigens. Shigella is non-modal, so it lacks the H antigens. Shigella sonii is the predominant species that causes human disease in the United States, followed by Shigella flexneri. Shigellosis is caused by Shigella sonii, which is a short, self-limiting disease that usually causes symptoms such as fever and a very watery diarrhea. Shigella flexneri was more commonly associated with disease in children, but it has recently been associated with infections in young adults up to 25 years of age. Shigella flexneri is the leading isolate of gastrointestinal disease among homosexual men, and in developing countries, Shigella dysenteriae type 1 and Shigella boydii are the most common isolates. Shigella dysenteriae type 1 is the most virulent of all the Shigella species and has a mortality rate of 5 to 10%. And humans are the only known reservoir for Shigella. Transmission usually occurs through person-to-person contact, the fecal-oral route, as most of these organisms are. Carriers can also be a source of transmission. Insects, fingers, water, contaminated food. So personal hygiene plays an important role in Shigella transmission. So transmission of Shigella is much more common in children in daycare centers and people living in very crowded conditions where you might have a lower hygiene level, especially in your daycares where you have these little kids that might be using the bathroom, not washing their hands, maybe, usually daycares don't have diapers, but kids have accidents, and so Shigella tends to be common in children in daycare centers in the U.S. Children less than 10 years of age are most highly affected, and those that are 1 year of age or less are more susceptible. Fewer than 200 bacilli are needed to initiate disease. So this has a very, very low infectious dose. If you remember, we talked about salmonella and the pathogenic E. coli. There were 10 of the 6 organisms are the infectious dose. So Shigella, you only need 200 bacilli. The mechanism of disease, again, Shigella causes bacillary dysentery, which is caused by the organism penetrating the intestinal epithelial cells after it attaches to the mucosal surfaces. It generates a local inflammation that causes shedding of the intestinal lining and the formation of ulcers. So people can get different types of symptoms. So they can have a completely asymptomatic infection, and they can also have a very severe disease. Initial symptoms usually are high fever, chills, abdominal cramping, and pain accompanied by tenesmus. And tenesmus is straining to defecate. Incubation period is 24 to 48 hours after ingestion of the organism, and usually you'll see bloody stools that contain mucus and a lot of white blood cells. Shigella dysenteriae causes a dysentery and has much more severe symptoms. So individuals will get a bloody diarrhea that progress into dysentery. It can last for a few days or just a few hours. Usually it's extremely painful bowel movements containing mucus and blood. The problem with Shigella is it can cause such an awful watery diarrhea and a lot of straining is it can generate rectal prolapses and also other complications such as obstruction of the intestines. We're now going to move on to the 5th part of our Enterobacteriaceae lecture.